to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 73rd episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO servicing the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Moreno, and I've been in the LBA and cell and gene therapy space for almost 30 years now. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for over 29 years.
John and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. I'm located in Kansas. John is in upstate New York, and our producer is in Missouri, so we're all over the map today. We are ecstatic to have you listening to the 73rd episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of our latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from any of you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. To kick us off for podcast number 73, John is going to go over today's podcast topic a bit more before we jump into the news and resources section. John?
So, as it's the first of the year and a happy new year to all our listeners, I thought it might be nice to have a bit of a recap about the parallel evolution of bioanalysis and how KCAS has evolved with that, and leading into a bit of an announcement about where KCAS is going for the future.
Ooh, a teaser, John. Well, with that, let's just jump right into the news and resources. John?
So, first topic, lutetium, actinium, or lead, can isotope type offer an edge in radiotherapy? Thought this is worth talking about because, you know, it's, if you watch what's going on in the industry, the interest in radioligand therapies is markedly increased. There's a couple of approved drugs on the market, but also I think there's a couple of Novartis therapies, but companies like Eli Lilly and Bristol Myers Squibb are sort of moving to invest in smaller companies who are exploring this, and it's something we've had some involvement in. And it's just, you know, it's similar to sort of ADCs where you're using the antibody to help deliver the radioactive isotope to the site of action, where it then decays and, depending on type of radiation, you know, treats the tumor cell in situ. So it's obviously it's an interesting concept. It seems to be gaining a lot of traction, and also for us, it's bioanalytically interesting as well.
Yeah, John. These radioligand therapies (RLTs) are certainly. The article kind of alludes to where ADCs were 4 or 5 years ago, right? So they're really
exactly
incredibly powerful therapeutics, right? Bioanalytically, they have some challenges. The names of these things are very difficult for me. I'm glad you're saying them. But no, you know, John, you mentioned a couple companies. Novartis is getting into this. There's a lot of, like radio labels are kind of taboo a little bit, right? But they're
absolutely
very powerful things. And, you know, as an organization, John, and we can talk a little bit more about it, but we're like, we've got our radio label back again. We're thinking about getting a scintillating counter because we can't outsource some of the testing because we're not a radio labeling. We're not like a radio label RIA assay shop anymore, John, right? When I first got here, we did have that platform. And so we moved away from it because of the sensitivity of some of the other ligand binding assays that are out there. But it's like anything else. It's kind of like fashion, John. It's coming back.
And for us with LCMS, you know, typically we don't have a need of a radioactive license. But when you have C14 studies, then you do need to be able to deal with those samples where they're going to have a hot component. You're only going to be looking at the unlabeled analyte, but because there's C14 in there, you have to have the ability to deal with the samples, then clean up afterwards, which is where we're going now. The thing about these radioligand therapies are actually using short short-lived isotopes, so they don't actually, the half-lives of the ones they're talking about, of course I can't
10
find
days,
it. It's in order,
10
yeah, I
days,
think
half
actinium,
days,
actinium 225 is 10 days, lutetium is lutetium 177 is lesser, and then there also is interest in lead 212, but lead 212 is the order of hours, so it's actually much more challenging logistically to administer. Where we've worked with these compounds, basically the samples have been, obviously the study's been done, then samples have been held until the radioactive level is down low enough that then they can be safely handled without a radioactive license. So there's a delay at the central lab before it gets shipped to us. It like say there's two camps. There's those with lutetium looking at beta radiation, which is more penetrative. Things like actinium 225 and lead 212 are alpha emitters, which I remember from high school is stopped by a sheet of paper. So it's less penetrative in terms of how far it gets into the body. But it's just, I thought it's a really interesting field. It's starting to get a lot of attention, and it just seemed like an appropriate time to start talking about it, particularly as there was an article I could lift from the internet to initiate the chat.
Yeah, so cold label studies, right? But it just seems like it's really coming back, and they're very powerful. And as I mentioned, John, we are in the process of when we moved, we discontinued the radio license, but it seems like that's coming back, and rumor has it we're getting a scintillating counter as well. So stay tuned there. But definitely, you mentioned high school and the alpha and beta emitters, man. That brought me way back, right? That's kind of funny, right? Like you said, a piece of paper, or I remember like I was taught a T-shirt could stop it.
Yeah, beta isn't very penetrative either. So I'm trying to think what it does say. What it basically. Beta emitters deliver electrons that can travel 2 to 6 millimeters and work well in targets with heterogeneous expression. They cause single-strand DNA breaks, which can be effective in stopping tumor progression and triggering cell death. So that's why the lutetium. For the alpha, the particles are big because that's effectively, it's like a helium particle, isn't it? The size of a nucleus versus an electron are more potent. They cause double-stranded DNA breaks, but they only travel 1 to 3 cells distance, so they have a very selective range, but can cause massive problems to cells. That's from the Fusion CEO John Valiant. So basically, both have their application, and I think it's probably down to ease of use, and like I say, the lead 212 is there. But I think the other piece of this, and that's why lead 212 is being discussed, is that both actinium and lutetium, I think, have a limited source as to where they come from, whereas lutetium is actually comes naturally from thorium 228, so it can be much more available in terms of production. So there is another component of this is the actual production of the materials has its challenges.
Oh yeah, you bet. And lead seems like such a taboo metal, right? Like it just lead paint, all that sort of lead. But,
actinium sounds pretty taboo to me, to be quite honest.
They all are, right? Yeah. Now that I say it out loud, but like
it's right up that far end of the periodic table where no one wants to go,
But if you said to the, if you're in the grocery store and you're like, oh, this has actinium in it, no one doesn't know what that means.
Exactly.
If it had lead in it, people are gonna run from it. But these are the therapeutic itself has that same, and such great therapies out there. They all got to get to the site, right? That's still the major.
Exactly, yeah.
And then like the delivery, it takes advantage of the tumor, right? Because tumors are sitting there sampling at much higher than your cells. So even though that small penetration of cells, those normal cells have a better chance of surviving if they are in the proximity of the radio label, because they're not taking it up at as high a rate, right?
Okay.
Even though they are sort of penetrating, but that's kind of the principle behind it, right? So it's really good stuff, and great way to start our first news article of the day, John. But it might lead nice to the next one. I definitely saw it hit my desk because. Any more thoughts on that one, or we want to move on?
No, we can move on. The next one is it's another thing that's in the news a lot, but this is actually getting more understanding of what's going on. So this is explainable AI finds new antibiotics that stop MRSA in mice. So this is twofold. I mean, obviously we've talked about this repeatedly about the concern of resistance of bacteria to therapies. So there's this constant look for new antibiotics to overcome resistance. And so this group at the Massachusetts Institute of Technology basically used artificial intelligence to really look for classes of antibiotics that could kill methicillin-resistant Staphylococcus aureus or MRSA. This is published in Nature, so this is their take on this was they used AI to do it, but they actually tweaked the AI to explain why it was going in a certain direction, so they could follow the logic of what was being done. So it made more sense to them rather than the AI just sort of going off and doing its things. And here's a compound, they could actually see what the process was, and it actually sort of helped them figure out new classes of small molecules that could be capable of like I said, dealing with MRSA. So it just seemed like an interesting, because to a lot of us, AI is a black box. Here's a group who are sort of saying we want to really get away from the black box and figure out how can we use it, but know what the reasoning process is to then really take things further.
Yeah, John. It's AI is all the rage, right? In the bioanalytical world, they're trying to incorporate it, but it still has a lot of I don't know if downside is the right word, fault, however you want to think of it, because we can't share data appropriately. So even if you try to plug in every data bit we've generated at KCAS or KCAS Bio, excuse me, it's a fraction of the data out there. Where this instance, right, they took it's mind-blowing numbers of small molecules, like 1 million or something, and they parsed it out, and so they kind of teased it down, and then I think they looked at sort of the antibiotics themselves, and so this is AI, AI at its finest, in my opinion. AI where this is to me, it's like sound science. It's helping to narrow the let's say funnel of compounds to treat MRSA. And again, we talked about MRSA. It's one of those things that we all should be a little bit worried about. But it would be great to have some, antibiotics that are available to just combat it. And they're on their way. And of course, it's MIT, right? So it's got all the bells and whistles of why America, why science is so great. Globally.
So it's here's the gist of the process. They started with screening a set of 39,000 antibiotics, natural products, and other molecules for their ability to stop MRSA from growing. This data was then used with a program called ChemProp to train neural networks to classify whether or not a compound was likely to inhibit MRSA based on its chemical structure, supplementing the models with additional data to improve accuracy. They built similar neural networks that could then predict whether the compound would be toxic to human cells. And then they engineered those models to explain not only which compounds are selected antibiotic activity, but also why in terms of structure, and that's where they can build from there. So you're right. It started with 12 million compounds, and that's...
Think about that for a second. 12 million, right, John? Yeah. 12 million small molecule compounds. That's unbelievable.
Well, I like the fact that I've actually learned a word today. Our explainable approach provides a blueprint for how the infinitude of drug-like small molecules can be efficiently mined for new antibiotics using deep learning. So infinitude there's a new one for me.
Infinitude and beyond, is that? No, sorry.
Yes. Not mocking it. That's always been the premise behind drug discovery. Is obviously you can have infinite number of structures that you could be synthesizing to come up with hopefully the therapeutic activity you're looking for. I mean, that was a big push behind combinatorial chemistry and drug discovery back in the late 80s, early 90s. Was just there was departments just dedicated to synthesizing small molecules because of all the different possibilities. So that way, I don't know if there's much more to say on that. There's anything?
No, just a wonderful story. It's all the things that are helping to advance science. AI, unmet, kind of a need that's bubbling under the surface. I'm sure every hospital in America would love something to combat MRSA if they could. Every locker room, it's not in the news as much, but it certainly didn't go away, did it, John? Right?
Exactly.
I feel like it's only going to resurface as we start to just kind of, open up more and more. But yeah, no, we can move on from that. I'm still trying to grasp the 12 million small molecule compounds, and even you mentioned the 80s, but like I think you could go back into like the early 1900s or 1800s when like Bayer is founded, and a lot of these are coming from like oil supplements and things that were from the wall, right? I don't know. I read a couple books about how they kind of were like they were burning whale blubber, and they're like, what's this residue on the wall? And then it was like killing bacteria. It's like, oh, what is this, right? That's kind of some of how some of that was discovered, not just penicillin and other things, but really I believe that is how some of this came about. Was just
and just as a footnote to this, this is anticipated or planned to go into the clinic, so they're looking to develop compounds from this.
Oh yeah.
With a company called Fair Bio, of which Collins, who I'm not sure what Collins, I missed that, is a co-founder. James Collins, PhD at MIT, is a co-founder of Fair Bio. So this hopefully could benefit people in the long term. So let's move on to another thing. You say it's a problem that hasn't been met. Obviously, we started this podcast in reaction to COVID and how it affected our daily lives. And for a lot of us, those of us had COVID, we've had brief flare-ups, we've basically dealt with it and moved on with our lives. But there's a significant portion of the population who haven't. Those who basically end up with long COVID, which has a multitude of symptoms and can be quite debilitating for people. But this is actually a group in Zurich, in Switzerland, who think they may have an explanation behind the mechanism for the mechanism behind long COVID, and hopefully that's the first step to thinking about new treatments. And this was published in Science. 39 healthy people and 113 COVID-19 patients who are followed for 1 year. Blood samples from the 40 COVID patients with long COVID contain signs that the complement system, part of the immune system's first line of defense against pathogens, never turns off after its encounter with the SARS-CoV-2 virus. So basically, once the complement system's triggered, it never actually turns back off, and so then it's actually then turning on your healthy body cells. So I'll pass it back to you, but I just thought this was a really interesting, hopefully first step to helping people, because it's really drastically affected people's lives.
Yeah, you said it well there. It's a early first step, and something that whether it's long COVID, even just understanding complement I've talked about this before. It's a therapy. You asked me about complement 10 years ago, I would have said, no way would I take a complement therapeutic. But they're getting bigger, better, and badder. I think there are some things that could help with complement. We work on a couple compounds. And so this first step, John, they talk about, and complement's incredibly, it's activation, it's suppression. All of that is still being unwound. I mean, we know there are 3 complement pathways. It's dense, John, right? Sure. If you looked at a complement chart, you're going to get to a point where there's more question marks as you start to. Now we know about what's called a MAC. There's like C5. It forms this killing of a cell. There's all sorts of things we know, but there are plenty of other steps in that pathway that are question marks, John. And in fact, the 3rd pathway is only about 15 years old now. So there might even be more unmet in terms of just what's involved in complement. So I want to set the stage around innate immunity. It's all pattern recognition. This is stuff it's just on.
Yep.
Right? It doesn't have like your immune cells got to go through like a wave of activation. Even some of your innate immune cells, they can be activated immediately, but a lot of them have to go through like a maturation process. Complement, on the other end, it's just like a light switch. It's just on, off, on, off, on, off. And so what they've discovered in this article very nicely is that, and we heard about blood clot formation. So all of those things are with complement, but it's TSP1, which we have an assay for, and the von Willebrand factor, which we've got another assay for. So these are things that we know near and dear. The trick, it's great they've identified it. This has got to bring a lot of peace of mind to.
So yeah, I mean, you mentioned TSP1 and von Willebrand factor. Basically, they're saying all of the patients with long COVID had markedly higher levels of proteins that facilitate interactions between the complement system and blood clot formation, such as TSP1 and von Willebrand factor. And then they also said they had lower levels of protein called ADAM-TS13, which cleaves von Willebrand factor and stops the blood clotting process. So yeah, that rolls into what you said about the blood clotting. There's they're pointing to this as some of the interaction that's leading to these blood clots that have been observed in long COVID patients.
Yeah, that's a very important factor you said, John, because just like tumors, you need the brake cut and the gas pedal on. So even though there is an elevated level of these complement factors, you needed to have something that was on the back end. So you needed a second kind of hit in order to have this long COVID. So it's a twofold thing. It's the lack of this particular protein, which is deactivating or suppressing the effect of the Willebrand factor, right? So it's all the redundancies. The body's powerful, and then there's one last thing too. The number of Epstein-Barr virus and CMV antibodies has an impact, which is all. It's just one massive orchestra, is how I think of it. And some of the instruments are a little off, and you got long COVID, right? That's kind of what goes through my mind.
Sure.
You know, the conductor is doing his thing, and you know a certain brand isn't triggering a certain section of the orchestra isn't performing at the right time. That's what this is, and it's how to fix it. That's part of what's happening. But I think for anybody who has long COVID who does listen to us, this is a wonderful go talk to your physician about complement and how you can try, because there should be some things to suppress that. Knowing is half the battle, right, John? Targets.
Yeah. So like I said, to me this is fascinating. I thought that it's definitely worth raising, and it's not often we touch on COVID, but this is something that definitely merited.
Yeah, well, it didn't go away. Everybody in my family's got it right now, John. I mean, my Colorado.
Oh, really?
Oh yeah, it's running rampant in Colorado. No one's like deathly ill. It's just but you know, what I would say is herd immunity is kicking in, but let's not lose fact. We should still try to distance yourself from people. Wash your hands. I'm not saying walk around with a mask, but if you want to wear a mask, wear one. But good Lord, we should not let some of the practices we had go away. But anyhow, I try about it. It's not, John, we've talked about this, right? Like all the COVID could fit in a can of Coca-Cola in the world. It's not gone anywhere, unfortunately. It's still ubiquitous. It's just we've become immune to it. But that's enough about COVID for now. I don't want to. How about it?
Sure. Yeah, I think it's. I think it has changed how we behave, and we definitely.
So. I see people cleaning more. I like that. I see a lot more. I feel like it's cleaner in restaurants. I hope we can keep that up.
Yep. So anyway, let's move on. The last one is just quick touch on investment news, or it's not so much investment, but really, as we move into a new year, we've had the biotech industry has had a rough couple of years with obviously with layoffs, just companies going out of business. After the where we were, again touching on COVID, where we were during COVID, it was almost like a boom time. Coming off that has been pretty rough for a lot of companies. So this was just a quick article in Endpoints News saying newly hopeful biopharma still faces an uncertain 2024. And it's interesting to watch what's going on. We are hearing there's been a number of fairly big acquisitions lately, but then Novartis there was talk of Cytokinetics being bought by Novartis, but then Novartis seemed to be backpedaling out to that one. Obviously, we've seen mergers. We're also seeing talk of IPOs with a number of companies actually announcing they're going public. But that's very different from 2023. But it's still, although it's there's green shoots of hopefulness, and this was mentioned at JP Morgan conference. It's still really uncertain what 2024 is going to bring. But I thought at least right now, new year, we're feeling a little bit more optimistic than it's been. But obviously, we're going to watch it closely to see how things go.
Yeah, John. I mean, it was 2023 was it. Everything has to have some down to it, right? That's just the cycle of life. Yep. So it was headwinds is the term we've used. Definitely saw a lot of layoffs. Biotechs got hit hard. It seemed like if you were a mature biotech, you were in good shape. The article kind of states that. There's some pretty fascinating numbers around 2023. 84% of all acquisitions were in that phase 2. and that's up from 2020. One, which was 68%. And then really the biggest thing that from 2023, and we can talk about 2024, is there were no preclinical IPOs last year. That struck me as like, wow.
Sure.
As opposed to like 25 of them 2 years ago. So from 25 in 2021, they don't tell us what the 2022 number is, to 0 in 2023 is that's all. That to me, that's a clear indication that we had this dip. JP Morgan, right? That just happened last month or earlier this month. Big conference. If you don't know that one, it's a who's of conferences. I believe our producer was there because he's a smoozer. But no, that one had big lift. There was some nice things coming out. All signs pointing the right way. Although there's been a little bit of regression in the market from that. But John, I mean, clearly, from our standpoint, I feel like there's been a pretty nice uptick, and we're just feeling more comfortable. And I want to end with, or at least pass it over to you after I say, like, it wasn't a lack of money. I don't think that was it. I think people just got hesitant, John, and they were spending less. And now it seems like it might open up a little bit. But, I'm optimistic about this year. Where last year at this time, you could kind of sense we were going to have a downtick.
I'm a naturally glass half full kind of person, so I'm feeling optimistic too. But yeah, I think it was people were being very careful with how they were spending, and it's sort of that was a lot of the story was, the companies were finding it very difficult to fundraise because like people were not investing. So the hope is that this, we're on that.
Yeah, the bank thing too, right? The Silicon Bank hurt, John, right? The rates went up. All of that made it.
Sure, absolutely.
That had an impact on 2020.
Yeah, sure. Yeah. So,
good deal.
Yes, let's move on. That's the end of the news. Let's move on to the main topic. So for this, we talk about the weekly bioanalysis, and we dip in and out of what we do. And I thought, we have some big news at the end of it, but I thought it might be just quite good to talk about the parallel evolution of bioanalysis and KCAS. So really almost go back to square one as to what is bioanalysis, how's it changed over the years, and then we've got a kicker at the end to just talk about. I'm still saying KCAS. I'm going to leave it.
That's okay.
We'll change it towards the end. But yeah, so that's all. I thought it might be just a nice sort of, like say, dip back into history, and then sort of say here's where we are going for the future. I mean, obviously as we've evolved with the industry, it just seemed like a good way to go on this today, as in the first of the year, So we can. I'll start off. I mean,
sure. No, we got a rich history here. We've been around since 1979. So and wherever you want to take us, John, I'm along for the ride.
Yeah, I'm not sure I'm going to start, but let's see. So basically, yeah, I mean, obviously we talk about this all the time. We're always talking about drug development in this podcast, and an essential piece of drug development is really, I'll start from the PK side, the pharmacokinetic side of things. You need to know if you're administering something to a biological system, what that compound is doing in terms of how high a concentration it's reaching, how long is it hanging around, because the things like the half-life of the compound, etc., are all really important parameters, because actually depends on what your therapy is. You may not actually want a long half-life. You may want to come in and out of the body quickly. For the ideal way that we can determine that is bioanalysis, which is where KCAS and a lot of other companies step in. What we will do is, I'll just talk my LCMS with my LCMS hat on. We will be asked to develop an assay for a molecule, so we will develop that method. With us it's extraction. I'll touch on this a little bit more. It's extraction, chromatography, mass spectrometry is the 3-step process. We'll develop that assay, we'll validate that assay, which is showing that really showing the reproducibility of the assay and things like stability. We're measuring stability of the analyte to make sure that when we get samples, we're still getting reliable measurement. And then once we are running a study, we'll receive a bunch of samples, and those samples will have been taken at either the clinic or like an animal facility at set times, and those samples will all be labeled with subject and time point, and so we will extract each of those when the LCMS we're extracting, we'll extract each of those samples, analyze those samples, such that with those set time points, we can build up a profile of what the drug is doing in the body. And that is the gist of bioanalysis. That's pharmacokinetics. That's you're administering a drug and you're seeing what it's doing. We also, because there's a bit, this is also a huge component, is the pharmacodynamics, which is you administer a drug and then you're watching the effect of it on known biomarkers, and that's another big component of bioanalysis, because that's same principle in terms of coming up with a method, but you're looking at a molecule or whatever that is already in the body and our effect of the drug on that in the body. So that's really bioanalysis. And like I say, this is all critical to drug development. So you know what your drug's doing in the body, you know what effect it's having on the body, and is that effect desired for that drug in terms of how our biomarkers behaving in a predictable way. And I'll pass it back to you, Dominic, before my mouth goes dry.
No, that's all right. I feel like the original pharmacokinetic experiment is to eat some asparagus, John. That's the, you know what I mean? Like there's, to me, that you can literally eat it, and then when you go bathroom, you can smell it. That to me is like, if you want to break down that's an experiment all of us have done that says, oh wow, the body does process stuff fast. So
yes,
that was a little crude, but that's, to me, that's the best way to think about how quickly your body can break something down, right? And you very nicely kind of went over the LCMS portion of things, and you got this extraction, you got this chromatography, you got the mass spec, and all through that you talked about the reproducibility, but it's like the specificity of it, the selectivity, robustness, yep. All of that plays into things. And up until maybe 1988, 90 range, that was what everybody was doing. It was all molecule stuff.
Yeah, if you want, I'll just give it. Yeah, I mean, basically that was. Yeah, that was it. So for in the early days, if you look at a lot of early papers, it's actually LC, because the MS piece hadn't been resolved. It was actually a real struggle to actually interface LC with mass spectrometry, and so. The reason why I ended up with a PhD is I was in a group who were trying to figure out how can we best interface chromatography with mass spectrometry so you have a robust path forward. I'll be honest, we went down some tangents that in the end turned out not to have the future. And it's really, you'll hear mention of electrospray. That electrospray is an ionization. So here's the issue with LCMS, and sorry to interrupt on you, Dominic.
Okay.
The fundamental problem with LCMS is liquid chromatography is a high pressure technique. Mass spectrometry is a high vacuum technique. So you've got two basically incompatible techniques that you really want to marry. So the solution to that was electrospray where where you're forming ions that exit from the LC column. You're doing that at atmospheric pressure, and then you're sampling from those ions into the high vacuum. So when we were working, our early days, we were all trying to introduce ions trying to introduce the LC effluent into the mass spectrometer and ionize within the mass spectrometer. That external ionization was the solution to marrying LC and MS. And then there's been various things added to make that more robust. Electrospray by itself wasn't really that robust, but adding sort of various gases to help control the spray, and then heat on the front end of the mass spectrometer also help evaporation, and then sampling in terms of how optics on the mass spectrometers. What this did was made LCMS it basically made the marriage work, and then the tweaks thereafter have made it a very robust technology, such that you have now this what is considered the gold standard for small molecule analysis. And most of the work that we do is done overnight when no one's in the building, because you can trust those instruments to do what needs to be done. And that's small molecule, and small molecule really, because the drugs that we were working on were all small molecule, that's why LCMS was really at the forefront of bioanalysis. class. So that's why it took off so quickly, and why it's a very powerful combination. Because when I talk about the 3 steps of extraction, chromatography, mass spec, you can tune all 3 of those to optimize your assay, and that's where the power of the technology comes in. But then as drug design evolved, then there was always focus on large molecule, and obviously this is where you come in, Dominic. Large molecule analysis. You said late 80s where it's taken off. I have no concept of the history of that, so I'm going to pass it back to you.
Yeah, and I'll just say whatever a triple quad is, John, because that has to play. But that's a whole nother. Don't.
Yeah, that's part. That's
Yeah.
that's the triple quadruple mass spectrometer is part of your robustness of the technology.
Yeah.
So again, I will touch it very quickly. Basically, early mass spectrometers. So there was magnetic sectors which were huge and have become obsolete. The next technology was quadrupoles, where you've got 2 opposite sets. You've got 4 poles, both 2 opposite. There's the 2 opposite poles within the 4 operate in tandem with each other, and through the tuning of the voltages, you can transmit ions through the quadrupole. When you get to a triple, because here's another of the nice pieces of mass spectrometry. At the triple, you have the first quadrupole, which is resolving what those analytes you're introducing into the quadrupole. The second quadrupole is just a cell where you're containing those ions, and you're then colliding them with a gas, because by doing that, you can actually fragment those molecules in a known fashion. It will be predictable, and based on that predictable fragmentation, then you'll get fragments that will go in. You'll then resolve the fragments through the third quadrupole, and by monitoring between the first and third quadrupoles, then you get a predictable molecular, say, molecular ion to fragment transition, and that's the basis of what we're doing on a daily basis. And again, it really increases the selectivity. You can do LCMS with a single quadrupole, but there's so much background, it's really difficult to get down to the levels that we need to get down to. That ability to do what we call tandem mass spectrometry, that's how you can get down to picogram per mil and nanogram per mil levels, and that's where you get your sensitivity to really accurately measure drugs in biological fluids and tissues.
Yeah, no, thanks. Actually have heard these types of discussions. Not recently, but that made a lot more sense. It's still a black box to me. It's still a little bit of a mystery, but that was helpful. And,
sure, yeah, I think as you grow up with the technology, you know it, but then as you say, that's a black box. Ligand binding is like the
ultimate. It's magical, right?
So it's just what you get familiar with and happy with, and yeah, I mean, I could talk about LCMS till I'm blue in the face, but if you ask me a question about ligand binding, I'll hide, so you don't talk to me ever again.
You don't want to know about what's on the MSD plates, do you? Right. So, and then I think, before I jump in, you mentioned the PD effect. That's an important piece of bioanalysis, John. That's sometimes overlooked. It actually so just because you can measure a PK profile, right? Like over a certain time, it peaks and then it goes away. That's all great, but if it's not working, then who cares, right? So that's where that PD piece becomes really an important part of early drug discovery. Just because you can dose someone with a drug and you can get it to cycle through the life of the drug the right, however you deem appropriate, because you can tweak all that by dosages and so forth. If it's not actually working, then who cares, right? So that pharmacodynamic piece is really important. So maybe I'll jump into the ligand binding assays, and they date back to the 60s and even into like the 50s. Yep. That's what people were just like, they kind of knew these antibodies. I mean, it even goes further back. If you heard the story of the milkmaid who's they didn't get anthrax because they got passive immunity from the cows and stuff like that. There's a whole series of kind of history where people kind of knew there was something, and not until about I think it was around the 50s that they started being able to isolate antibodies, and that really that's when they started to put an antigen down, throw a serum sample in, the antibodies bound to it, and then they found a way to detect it. And if that kind of has evolved, and fast forward into, say, drug therapeutics themselves, That wasn't until like 1980, I think, is when they isolated the first monoclonal antibody. So they're kind of using like polyclonal sera, right, John? If you know what that term is. That's like you could just take the serum from us, and they were using assays. They were all sorts of creative ways to kind of look to see if you had like antibodies against a certain virus. There's all sorts of things they were kind of doing without really truly understanding, because they didn't have things like electron microscopy or some of the tools we have today to kind of see what was really going on, or some of the isolation techniques that we have to isolate. And then, of course, like the advent of hybridomas and the generation of monoclonal antibodies, that was a major advancement. And that's like its own little story. And again, that's all in like the 70s and 80s. But really, like in the cover of Time magazine, I think it was like 1990, it was like, is this the magic bullet? And monoclonal antibodies kind of hit the market. And a ligand binding assay, there's a couple different ways you can do it. One is to put your target down, and that's like the antigen down, whatever protein you're trying to detect. And then you come back with an antibody that binds to it. And sometimes that can have an enzyme on it that can cause the colorimetric reaction. Or you need a second antibody that has the colorimetric enzyme on it, because it's a color change, right? It's all fluorescence and mass spectrometry, right?
Yep.
And so then with that those have sensitivity and their specificity and robustness that you can optimize them. But the capture or sandwich antibodies is a sandwich ELISA was really what is the mainstream of what we do today. It's what the MSD instrument utilizes. And that's where you actually put an antibody down against the target. Then you add the sample with the target in it, and you come back with a second antibody. So they're antibody pairs. Now, ideally, those pairs bind to different regions on the protein. That's not always possible, big investment. They can be against the same region because there's like Brownian movement, and there's a whole piece of real estate on there that the antibody binds to. But if you can get separate regions of the protein, you can make an assay that is very specific, highly sensitive, highly robust. And so that's like the golden investment, is to make antibody pairs. Not always doable. You've got all sorts of complications in there about what's called discontinuous and continuous epitopes. It's the way the protein folds, right? So sometime an antibody can bind to like amino acids, say 200 and 251 or 200, 201, and then the other arms bind into like 400 and 420 because it folds a certain way. See, right? Yep. That's a discontinuous epitope versus linearizing the protein. So there's all sorts of science and engineering behind trying to do some of this. But, I hope that's a little bit of a walk down ligand binding assay way. But now we can, over the last 10 years, the ability to make monoclonal antibodies, we can make them against small molecules now, John, right? The size, the specificity of them, the piece of real estate, that's what I think of it, right? If you think of amino acid sequences, they can target things, where we can get to a point where if you put enough effort into it, you can tell a recombinant protein from a mutant, or like an endogenous protein from a recombinant one, or an endogenous protein that's wild type versus the mutant type, all with an antibody. But hybrid's a better choice for that, because you can pull down it and look at it a little bit different, where if I got to detect that wild type versus the mutant type, I just told you I want two different pairs of antibodies, right? So it gets a little bit harder. So I hope that made a little bit of sense, and at least from, for me, those antibodies I still dream about them and still see them at night because I'm a nerd like that. But no, the whole, and then of course, when you think of the pharmacodynamic part of it, it's not just ligand binding assays and small molecules. It's also like, that's getting into the flow piece of things and cell-based assays, but there's still plenty of pharmacodynamic methods that are on mass spec ligand binding assays. But more and more, we're seeing some more of that, cell-based stuff. And it's not just the emerging technologies part of things. It's also like, hey, if I got a really potent small molecule inhibitor of a receptor, well, now I want to maybe look at the occupancy of it, right? That's a flow method. It can be done without flow cytometry, but it's becoming a little more mainstream. So I'll let you jump back in, John, but that's a quick walk down some of the bioanalysis. That's just the PK piece, John, and we're scratching the surface at the PD piece. And of course, on the large molecule space, we've got this wonderful thing called what, John? Immunogenicity, right?
Yes. Touched on that. So yeah, I remember we had an assay. We developed an LCMS assay. This is my early days at my previous company for LT4, and I think it was.
Leukotriene 4, which is that is a promiscuous pathway. The are headaches for ligand binding assays anyway.
And so, yeah, there was a lot of work done on it to get the sensitivity that was being asked for, and it was, I think it was tens of picogram per mils on a SciEx API3, so an old instrument. It was a lot of work went into it. We had what was a good assay, and I drink so much tea that I was used as the blank matrix because my urine was clean of LT4, so it was in urine, and so we had this assay and we got it validated, and it came to we quote for the sample analysis, and the client said, "Oh no, we're going to do it by immunoassay because it's much cheaper." And it's like, what? So that was our first instance of where ligand binding sort of took some work away from us, which
usurped you. Right, that ligand binding assay started to usurp.
There is a lot to that, because I think it's it's like LCMS people, we're so full of ourselves in terms of, we've got the gold standard, etc. And we're never happy that we're just constrained to small molecule work.
Oh, John, us ligand binding assay people have plenty of ego.
So there's always been this push. I remember the previous company, how can we get into large molecule? What can we do here? And of course that does feed into the hybrid methods for working in complement to LBA, because like you say, LBA is there, it can deal with so much large molecule stuff, but LCMS working with LBA gives you the complete toolkit to go at large molecules. And I've touched on this before, but ultimately the challenge with mass spectrometers is they only have a range of a few thousand. When you're talking large molecules, you're talking tens to hundreds of thousands molecular weight, which theoretically should be beyond our reach. However, there are two things that can help us here. Electrospray, as the ionization technique, multiply charges large molecules. So if you look at something like 10,000 molecular weight, you put 10 charges on it, it's 1,000 mass to charge, which is in the range of our mass spectrometer, so we can measure that directly. When it comes to something like 150,000 molecular weight, like an antibody, then we're unlikely to go to it directly. What we do is we basically steal from the ligand binding approach by using antibodies to pull our say our antibody that we're after out of the biological fluid. Then we digest with an enzyme, be it trypsin, chymotrypsin, lyse C, whatever. And what we're then looking at is a peptide that should be unique for that antibody that we can then track through our typical LCMS processes. And actually, a lot of our selectivity comes from that LCMS end of things. It's back to the what I said earlier. We have so many different steps. We can tune all those steps to give an assay that gets you to what you need. Yeah. And that's why hybrid comes in, and that's why it has some versatility. I'm always going to be an LCMS person, but I know it's never going to take over from LBA. But it really, the two of them are such a good complement to each other. It gives us the ability to solve a lot of problems for what people are interested in. And also, and the reason why I put this, what we're seeing is more and more complex therapeutics, such as tri-specifics, biospecifics, ADCs, all these pieces. We need the multiple tools to solve those problems, because it's obviously we got things like we talk about peptides are one beast, but then you've got the acylated peptides. You're adding a fatty acid onto the peptide. That changes the nature of the molecule. That has its own challenges. So we really need those two sets of tools to be able to measure both pharmacokinetic and some of the biomarkers that we're after.
Yeah, and then the packaging of them, all sorts of things around the drug delivery systems themselves have application for mass spec versus LBA. And then I think one of the funny things is even as you're sitting here and you mentioned leukotrienes, I think Dawn Duffield is somewhere going, what are they talking about? Because she spent a lot of time researching that. And I'll say one of the powers of mass spec when it comes to dense things like leukotrienes, because LTB4 has like 4 isotypes to it, right? So a ligand binding assay kit, you're usually just measuring total LTB.
Yep, sure.
If I want to know, that's where the hybrid or mass spec really they can start to look at multiplexing. So you can start to, instead of just you can get a total number, but you can also get some of those isotypes kind of laid out.
Yeah, it's that whole
a really powerful tool for if you need that type of I don't know, information, right? That if that's the context of use of the assay. But leukotrienes are near and dear to a lot of us here. And as I mentioned, Dawn's an expert in it. And I'm sure she's like, what are you talking about? So
we can talk about lipids another time. I'm sure we could have,
and maybe
why we wouldn't.
I think I want to be sensitive to the time here. We haven't even really touched on like immunogenicity. I guess we can just shelve that for now. But where do you want to take us, John?
I think it's just important. We talked about all this, the evolution of the technologies that basically this is where bioanalysis has gone. But KCAS has grown all the way with this in the future. KCAS has grown with this all the way from 1979, where it was LC based. It's then implemented ligand binding technologies, and more recently, this is where we move into flow and molecular technologies. And again, this is for me; these are boxes that do stuff that I don't understand. I'm more of a chemist than a biologist, much more of a chemist than a biologist, and this is much more on the biological end of things. And I'd love to sit down and have actually sit side by side with someone doing what they need to do to get more of an understanding. But yeah, to me, it's where a lot of groups are going, because it's learning more about what the drugs are doing. But that's me putting it at a very high level.
And so it's a good segue into maybe something here. I think it's almost 13 or 14 years ago is when ligand binding assays started at KCAS, and then flow cytometry offering started about 10 years ago now. Mm-hmm. But they kind of go hand in hand, right? You can't do a flow assay without antibodies. So a ligand binding assay just goes on an ELISA plate reader, or some sort of molecular device instrument, some sort of plate reader, does some sort of spec reading, and then they have the MSD and Luminex. So there's been some advancements in that space. With the platforms themselves increasing sensitivity, and MSD is maybe 15 years old now. Luminex, similar time frame, and allows to multiplex, right? ELISA can only still look at a single analyte. Multiplexing of different analytes is a very powerful tool for biomarker analysis. But flow cytometry has been around since the 80s as well. But it's all antibody dependent. So when you think about troubleshooting a method, troubleshooting an LBA method and troubleshooting a flow method, the front end of it, the actual, let's say, laboratory portion of it, the hands portion of it, kind of similar, John. But then, one is a plate reader. You can do some curve fitting. You can maybe change. Typically, you read at 450. The readings you can kind of mess with that a little bit, but you're limited. Now, on the flow side of things, that's artwork, right? You've heard me use that term before. That's where gating and that's where I would say our expertise lies in that we have individuals that know how to get that mass spec to hum the right way. Just like I'm sure running an LC kind of has that, or at least the mass spec has that to it. So very cool stuff though. And then, as an organization, we've brought on the hybrid system about 6 years ago now, right?
Yep.
So what the latest is our high-res mass spec. That's what we're going to bring on this year. And who knows, the next qPCR a couple years ago now, about 2 years ago. And then we'll bring on next gen sequencing probably the following year. And then someone will pop up with a new instrument and there'll be something else for us to do. But after we get next gen sequencing, I think we can make, if we get the high res, the next gens, I think we can make the claim to fame we could do a PK assay for any drug on the market, John. I think I've been fighting 11 years to make that statement true, and we're getting closer every day.
And I think as an analytical chemist within a CRO, that's exactly the claim you want to be able to make. You're walking in front of when you're talking.
It's not bragging if you can do it, John. It's not bragging.
No, absolutely not. Not at all. And also the other part of that is not only have we built this in Kansas, and this is why it leads into the final topic, we obviously absorbed Flowmetric in Pennsylvania, and then Active Biomarkers in Lyon, France. But as a result of that, we were basically three companies, all with the same goal, but all three different names. So part of what KCAS has done is talked about what's our identity going forward, such that all three companies now have the same name in common. So I will let you take it from there.
Yeah. So John, right, we're a global organization, right? And getting to that point where it was time for us to harmonize. We are harmonized. We've been working a lot to get SOPs the same, IT's the same, forecasting's the same, John, right? Revenue recognition the same, scheduling's the same. That's our initiative in 2023, right? We didn't buy anybody. We spent a lot of effort. There was a lot of I'd say effort, energy, time, investment in harmonizing our organizations, and proud to say to you that they are, right? We have shared SOPs, everything's the same. And then wait for it, John, right? I think drum roll, please, right? We're rebranding, right? Right? And instead of being KCAS Bioanalytical and Biomarker Services, we sent an email out last week and we're KCAS Bio. A lot of people call this KCAS anyways. And if you've ever looked at our email address, it's always said KCAS Bio. So it was a natural progression. We're rebranding ourselves. It's so, John, to be called KCAS Bio is awesome. It's really easy. It's not a completely new name, but it's a reboot. And boy, we got a lot of things that we're going to be rolling out this year to kind of promote ourselves. So if the competition's listening to us, look out. And if those of you that buy from us, stay tuned. We're going to have a lot of fun this year. And those of you that don't know us or might be listening to the first time, KCAS Bio is going to kick some butt in 2024.
That's the plan.
Yeah. No, John, it's an exciting time, right? I mean, it's a lot of effort. If you want to go to our website, we've rebranded the website. You can see the amount of content that we've put on there. It's a lot of we want to share what we're doing with the community. We want to help the community. I mean that, John. That is a lot of our culture. That is a lot of our mission. We like to help with life-saving drugs. You can go make money doing anything, John. You could sell Lamborghinis if you wanted to make money. But it's such an altruistic mindset of our organization. The love of science, the love of helping cure people of diseases. And stay tuned. We're going to have some podcasts with some individuals talking about rare diseases and how it's affected them. I think it's important for us to emphasize just sure, if we like a paycheck, John, but that's not why we get up in the morning. Sure. Our organization gets up because we love to help solve bioanalytical problems so that life-saving drugs can help patients.
Absolutely. But yeah, I mean, just to roll back a bit, I think we talk about harmonization, and there's been a lot of effort in that, and I know it's vital if you've got multiple sites to harmonize processes, but it is really important to have that single name such that you know that there isn't that then confusion for both the internal people and for customers. So to have a name that you all rally behind is really important going forward. But yes, I other than that, I agree with everything that you said. I mean, for me, it's all about what can we do to help?
And we're a couple minutes talking about this stuff, John. You know what I mean? When you're talking about mass spec, I feel like you could do a whole podcast just going over the history of mass spec, right? And then, for me, I jump around a little bit more because I kind of love to dabble in a whole bunch more. But I assure you, I could sit here for 5 hours and talk about binding assays, molecular flow, right? Cell
Oh, sure. Yeah, absolutely,
man. Yeah. So, John, I don't know if you have any more. We can.
No, I think it was. I just thought this was a nice way to kick off the new year.
Yeah.
It's certainly a lot of it is touching on things we've talked about in the past, but it never hurts to remind people what this is all about and why we talk about bioanalysis and why it excites us. I mean, this is all about, for us, it's like we know we're a vital cog in the drug development process, and we love being that vital cog in helping drugs get to market. It's really satisfying when you read the FDA list of approved drugs for you and thought, yep, I had a hand on that one, I had a hand on that one, I had a hand on that one, and that's what we're about.
Yeah, we don't research them, right? So a lot of times some of my graduate school friends are like, how come you never got into research? And it's like, you know why? Because what you just said there, John, when I watch a TV ad, I'm telling you about half the drugs on the market we've had some hand on or I've had some experience with. And then, of course, I sleep next to a scientist. So she's like, oh, we're making that one, right? Like, it's just it's mind-boggling, the D side of it. And, the research side has way more failure to it. Not that the D side doesn't. But, we work, if you think about it, I believe, 30% of all phase 1 drugs move into phase 2. There's a percentage of things that we're touching that are going to get approvals. And Sean, you've been working here 5-6 years, I think, and I'm up to 11. Man, we have a whole spattering of drugs on the market that we're like, oh wow, we did heavy lifting for them or solved a very big bioanalytical problem. It's so satisfying.
And actually, it's really funny. Last year, there's at least 2 drugs that were on. I mean, they were, I think, almost killed multiple times, but still got through to approval. There's it's I'm not going to name
That's
them.
hard. It's hard not to say it, right? You're like, oh crap, I'm going to breach some NDA or CDA. We're both stuntmen. I feel like we're the stuntmen of R&D.
There was one I worked on years ago, and it sort of seemed like it was hopeful, and it died, and it's like it's gone, and then all of a sudden it popped up, it's approved, and then another one that had a very checkered career, going from hand to hand, and went through multiple companies, but eventually made it to market. So it's really hard. I think that's the difference for us. We get our hands on so many different drugs. I know people in large pharma who've been there for decades and have never worked on an approved drug because it all died. They all died, and they just didn't. They were just unlucky in terms of the teams they worked on. And that's the difference. Whereas there's a good chance that because of what we do, we'll there's a lot of drugs in the market.
And you got to have a mindset that you got to let it go too, right? Like
sure.
Often, like any analyst I talk to, I'm like, hey, just remember, you're going to work on this hard for about 3, maybe 6 months. And then like it goes away, like huh?
Yep.
Like that's kind of, and then it might come back again when it gets to the next phase. But you got to drop that and move on to the next one, and that can be hard for people because they want to invest so much time in it. So it's a nice
sure. Well, like you say, those two that got to approval last year were very pleasant surprises because I was convinced they were toast.
Yeah.
But it's all you always keep these names in mind just in case, because I've one of those minds that sadly remembers numbers more than I should. So I still remember a lot of the drug numbers that I worked on and developed. And then you sort of say, I know that one. And I mean, there was actually one, and you sort of, I think I know that. Then you do some digging online, you find, it is one that you worked on previously.
I agree with you. Numbers and then song lyrics, John. For some reason, I remember the most random song lyrics or lines from movies. And as my son's getting older, he's watching some of these movies and he's like, you didn't make up that word. Like I used to say, you suspect, if you know the movie Good Will Hunting. Mm-hmm. I'm like, you suspect. And so we watched it for the first time and he just thought I was crazy talking that way. He's like, oh my God, that's Ben Affleck from that movie. I'm like, yeah. So sorry, random thought there, John. But yeah.
I think we should probably wrap up.
Yeah, no, this has been a fun, I will say, before we get into maybe our weekend things, and I don't know, it doesn't seem like we got any feedback, but we do have a little teaser for the next podcast. It'll be on rare diseases. We're going to have a special guest. It's a colleague of ours, John. We'll reveal her. I'll give you that when she's on, but she's going to talk about kind of why she got into what she's doing. And
that would be great.
Really, and it triggered a little bit for me, like some of the things that moved me towards science was some of my dad's ailments and things like that. If you think back to some of your early years, I believe you had your dad was a professor or something that influenced you. For me, it was a lot of it had to do with some of the sickness my dad went through and some of the frustration I felt through that because of lack of answers at the time. Mm-hmm. And so that kind of, it really did, it helped that I was kind of good in science, had people telling me get into science. I was actually a diplomatic relations major, John, if you didn't know that. And I started at Stonehill College because I spent a semester in Spain. I was a diplomatic relations major and walked up into the office and they had a list of all what your careers could be. And I was super excited. And I walked over to diplomatic relations and it was like one 8x11 piece of paper and it had like, you could be a linguist, a professor, I'm like, oh my God, what the? And then I looked over and there was one with like 8 different things, pages. I walked over and it said pre-med. So I walked over and switched my major, John. That's how I got pre-med. And the woman looked at me, she goes, oh, you can't just walk into pre-med. You got to have all this. And I'm like, well, I think I took all those things. Of course I had. I took every AP course in high school because that was like bragging rights. That's kind of why I did it. But little did I realize that set the foundation for me to be able to just switch over to pre-med. And the rest is history. I started taking my first biology class, my first chemistry classes. I just knew it was my calling. I'm like, oh, this is it. This is what I want to do.
Sure.
John, let's talk a little bit. You go ahead.
It's amazing how serendipitous it can be in terms of what the little thing that triggers your path for the rest of your life. I mean, I can touch on that another time. I'm not going to do it now.
John, for me this weekend, John, I don't know if you've heard, but the Chiefs are playing in the AFC Championship game for the 6th consecutive year. But there's something special about this weekend. It's not in Kansas City. The previous 5 years it was. So there's a lot of electricity here in Kansas City, getting all sorts of And plenty of parties we might be able to choose it. Not that I'm that popular, John. I don't want to sound like I'm super popular, but there's lots of things going on, and the city's electric. And I just feel like Pat Mahomes has got something to prove. He's 3-2 in these games. He's chasing Tom Brady to be what he considers the GOAT. Everybody feels like Baltimore's just going to roll him. I think the Kansas City Chiefs got a different plan.
No, it'd be interesting to watch. I, however, will. What day is that?
It's Sunday.
Sunday. Yeah, I'll probably be traveling at that point. So anyway, yeah, tomorrow I and my wife will be going down to New York City because one of my Christmas presents was tickets to see Dara O'Brien. He's an Irish comedian. Really, I love he's got quite a clever sense of humor, very warm. I love his presentation. So we're seeing him in a theatre. My eldest son is also joining us, and his wife, and so Thomas and I are going to see Dara O'Brien at a theatre on 43rd Street. So then we're spending the weekend in New York.
Oh wow!
Actually was on my phone yesterday, and I think it was Twitter, and it was Dara O'Brien flying into New York, showing a photo of the skyline as obviously the plane's turning round to land. So it was nice to see like him saying, "I'm coming into New York," and we're like, "Oh yeah, we'll see you there tomorrow." So yeah, it's that's our weekend.
Well, that's awesome. That's despite my jumping in with the Chiefs and everything, going to New York City is. That's a special moment. I love New York City, even though I don't like the Yankees. It's okay.
And the nice thing is, because we live near Ithaca and Cornell University, there's a bus that goes. I know I've mentioned this before. There's a bus that goes directly from Ithaca right into the centre of New York.
Oh, really? No stops? Like a zero stop?
No stops. Zero stop bus. It's excellent. So we have that service, and actually, I also have New York coming in. Was it 2 months' time? I don't know if you've heard of Orbital. Orbital, I think they started the late 80s. They're all involved in the rave scene. It's two brothers. It's like electronic music, but they've just done a reissue of their first album, and they decided to do like a tour based on the first album, and then they also decided, no, let's do the first and second album, and let's premiere that in New York. And so they're doing a show at Webster Hall in New York, which is the first and second album. That's the early show, and then they're doing a late show, which is their music festival set. And I'm like, I want to go, but which do I take? So I've basically bought both. So my son and I are doing Orbital in 2 months' time. Wow. Late, and so the late show doors open at 11. I've no idea how long I'm going to stay awake for that. But
vodka Red Bull, John. Vodka Red Bull.
Too good an opportunity to miss. So yes. Anyway, that's our New York stuff, and then that's our weekend, and honestly, that's about as far as I can think ahead at the moment.
Yeah. No, so. You've got that's fabulous stuff there, John. You're making me feel like I got to be a bit more adventurous, meaning it's finally going to get warm enough here. I might be able to break open my green egg again. And I can't do a podcast without mentioning pizza. And the problem now is, I think I mentioned this to you, my son works at a pizzeria called Minsky's, and he's there Wednesdays and Sundays. I couldn't be more proud of my son. He's doing unbelievable things. He got accepted 3 offers to his top choices for fraternities, because when you go into KU, you don't actually have to. Freshman men can go straight into fraternities. So he's been doing all sorts of activities there, and got 3 offers from the 3 he wanted to go to. So he's got some decision making to make there in the next coming days, but he thinks he knows where he's going. Just really that's great. But back to the pizza piece, he's bringing pizza home like Wednesdays and Sundays, and they make great pizza. So I haven't made a pizza in a while, and I'm kind of getting antsy. So I don't know. I might break open my Uni pizza oven, or at least make some wings on my green egg. We're supposed to get maybe 50-ish. I don't really like to cook outside until it gets over 50. But yeah, John, enjoy your safe travels. Enjoy that trip. Have a lot of fun. Vodka Red Bull's the way to go, in my opinion. But yeah, close us out, John.
Yep, absolutely. So that's it for us. Thanks everyone for listening, and look forward to the next one where we'll talk rare disease.